Hello everyone and welcome to this EPTRI webinar. Sorry for the delay. And first of all, I thank you for taking time out and being here today. I'm Angelica Intini, Innovation Project Manager at CVBF, and we are organizing this webinar in the framework of the European Biotech Week 2020. A week-long series of events celebrating biotechnology that take place annually around the month of October. The first European Biotech Week took place in 2013, marking the 60th anniversary of the discovery of the DNA molecule back in 1953. The webinar is aimed to raise awareness on the serious lack of medicines for children in Europe and worldwide, and to promote the application of biotechnologies to bring innovation in the pediatric drug discovery and early development phases. During the webinar, after an introduction aimed to describe the pediatric drug development context with the focus on EPTRI, the European Paediatric Translational Research Infrastructure, we will be looking at innovative approaches available in the pediatric research field through lectures given by experts in different fields. And we will conclude talking about the collaborative efforts to promote innovative drug discovery for children. Okay, now I just want to give you a few instructions on the webinar platform. There are 4 buttons in the top left-hand corner of the webinar panel. You can use the first button to expand or collapse your control panel, so to make it smaller or larger as you prefer. There are then 3 sections in the middle of the webinar panel. The audio section to change and control your audio preferences, and the question section where you can type your question or comment, and we will be running a question and answer session during the webinar. So if you have any question, just write them in there whenever you want during the speaker presentation. All the attendees will be mute for all duration of the webinar. And if you miss anything, don't worry. We will share the webinar recording on the EPTRI website in the events section. Before starting, I just a recommendation for the presenter. I kindly ask you to stay within the allocated time in order to avoid delay. Thank you and enjoy the webinar. I would like now to introduce today's first presenter, Donato Bonifazzi, CEO at Consortium per Valutazioni Biologiche e Farmacologiche and EPTRI Coordinator. Donato will give us a presentation talking about EPTRI, the European Paediatric Translational Research Infrastructure. Please,
Thank you,
Donato,
Angelica.
the floor is yours.
Thanks a lot, Angelica. Welcome, everybody. So I will go directly to the presentation in order to save time. The ID-EPTRI project was funded by the Horizon 2020, coordinated by Consortium per Valutazioni Biologiche and Farmacologiche, and aimed to design the framework of a new research infrastructure able to accelerate the pediatric drug development process with 29 partners from 21 European and not European countries. The main outcome of the project has been the production of the conceptual design report, describing the scientific and technical requirements as well as the key components of the new research infrastructure. And to prepare the conceptual design report, the project encompassed 3 phases. During the context analysis phase, the perceived value and the possible gaps to be covered have been estimated by inquiring the scientific communities, the concerned national authorities, and many other stakeholders. During the operational phase, the different components of the new research infrastructure have been planned, including governance model, strategies for interaction with the national authorities and the existing research infrastructure, the IT architecture model, services to be provided, and a business plan. Finally, a feasibility phase has been run to develop virtual exercises simulating the operations of EPTRI to work as a one-stop shop for advice in pediatric drug development. One of the main results within the context analysis survey showed that more than 300 research units from 29 countries are still interested to provide services in the framework of EPTRI for fostering pediatric research. So let's go a little back to understand the rationale of this new research infrastructure, which is proposed as a new infrastructure dedicated to pediatric research and aimed to cover some critical gaps that we can summarize easily as lack of integration, lack of funding, and lack of permanent supports. We will have a look to the different phases of a research infrastructure now, according to the life cycle as defined by ESFRI, that is the European Strategic Forum on Research Infrastructure, and which foresees 6 phases. They are concept development, design, preparation, implementation, operation, and termination. Until now, EPTRI has already concluded, of course, the concept development, which started several years ago with several projects that anticipated, European funded projects that anticipated the development of this new research infrastructure, and now has also concluded the second step, the design phase, which is aimed to test the scientific concept and technical feasibility of the future research infrastructure and the analysis of the potential user community. So, which is the EPTRI concept? To cover the wide needs of pediatric drug development research, it is necessary to aggregate all the available resources and to integrate all the competencies in a common effort. EPTRI aims to lead pediatric research to innovation through aggregation of a large research community focused on preclinical and translational research for pediatric medicines, making available innovative methods and technologies specifically developed for pediatric medicines, or even modified from what already exists for adults, and underpinning pediatric clinical research by translating basic preclinical and translational knowledge into clinical studies, design, and conduction. We have also worked in order to, during the design phase, in order to integrate EPTRI within the ESFRI landmarks, because there are several other biomedical research infrastructure. So in designing EPTRI, the relationships with other existing ESFRI research infrastructure have been carefully considered. In particular, 8 biomedical landscape research infrastructure have been identified as relevant with respect to some of the objectives of EPTRI, and they are BBMRI, EATRIS, ECRAN, Elixir, EU-OpenScreen, Eurobioimaging, InfraFrontier, and Instruct. And also a survey has been launched to investigate the availability of pediatric services in the portfolios of these 8 research infrastructure. We have concluded that relevant services proposed within EPTRI have never been developed in other research infrastructure, and that some basic research activities are not of interest of other research infrastructure, as, for example, research on human development mechanisms relevant for pediatric diseases. This is why the conclusion was that a new research infrastructure dedicated to pediatric research was extremely needed. So going in the details of the context analysis results within the conceptual design report of EPTRI. We can say that we have performed many actions, so 3 different surveys, and they were in the first one, in the context analysis survey, as I already said, more than 300 research groups in preclinical and translational research for pediatric drug development answered to the survey, declaring willingness to provide services within EPTRI. But in addition to it, an online survey has been conducted to identify the nature and the extent of the needs of the users for services in pediatric research, addressing the potential user community composed of scientists and experts, sponsors and contract research organizations, preclinical researchers and clinical investigators. And also within this survey, 317 potential users declared interest, and they also declared 2,200 pediatric research projects to be developed in the next 3 years. Finally, a third survey was a questionnaire circulated to all the stakeholders, like research organizations, but also academia, hospitals, pediatric networks, pharma companies, patient associations, regulatory agencies, and national authorities to investigate their agreement with the main concepts of EPTRI, its services, and also to provide comments and suggestions. Here we had a total of 155 questionnaires from 31 countries, including extra-European countries. So the next slide summarizes the architecture design of EPTRI with all its main components, with users and stakeholders, and also the other research infrastructure outside the EPTRI domain, while EPTRI in the center of the future will be established as a distributed research infrastructure based on a hub and spoke model with a central lab and several spokes. The research units identified during the EPTRI context analysis will be aggregated according to both geographical distribution in national nodes that you can see in the bottom of the figure, the national EPTRI infrastructure, and in scientific fields of activities that you can see in the center of the figure, that are the thematic research platforms. You can also see the central lab where all the activities are coordinated and managed, and the services are offered to a single access point, and also specific advisory boards and governing bodies are established. Through the integration of research organizations in both national nodes and the thematic research platforms, as described, EPTRI will be able to offer high-value services to the scientific community that will keep pace with evolving technologies and procedures, and that will be aligned with the demands of the pediatric community, so to follow innovation. The services that will be offered to users to the single access point will be three types of services. The integrated services that will be provided by the research units organized in the 4 thematic research platforms. And also an additional medical device platform is under development. This is to be mentioned because there was a very huge interest demonstrated during the EPTRI surveys. Then we have the centralized services that are managed and provided at central management office level, provided by this office together with experts from several EPTRI partner institutions. And finally, the common services provided in collaboration with other research infrastructure. What is also to be mentioned that in the EPTRI design, the results of basic and translational research are finalized to underpin pediatric clinical phases and to support the final use of age-tailored new and innovative medicines. To this aim, EPTRI offers also centralized services in agreement with relevant pediatric clinical initiatives, and mainly C4C, that is the Pan-European Network for Pediatric Clinical Trials. Now I will follow a quick description of the 4 main EPTRI thematic research platforms. The first is Pediatric Medicine Discovery, which is aimed to identify druggable molecular targets to pediatric diseases. Services within these platforms include implementation of in vitro, in vivo, and in silico models reproducing pediatric diseases, and in silico prediction of properties and toxicity for new molecular entity of pediatric interest. We have the developmental pharmacology platform consisting on the identification and application of ad hoc innovative methods to study changing pharmacological characteristics in the developmental population, such as PK/PD and PK models, in vitro models to study the ontogeny of drug disposition, modeling and simulations approach, and microdosing. Then we have the Pediatric Biomarkers and Biosamples platform, that will deal with the identification, characterization, and validation of pediatric biomarkers, bioinformatics for the analysis of data, and management of pediatric biosamples and related data. And finally, the Pediatric Medicines Formulations, which is aimed to perform age-tailored preformulation and formulation studies, covering the different subsets from preterm neonates to infant and adolescents, identify innovative alternative route of the administration, and perform delivery studies adapted to children's needs. Second type of services are the centralized services, which will be advice on translation to clinical phases, document repository and e-library, advice on the design and requirements of not clinical specific studies and experiments, access to learning and training, and also scouting of and application to funding opportunities with research project management in case of approval. Then finally, we have the third type of services. So the common services will be provided in collaboration with existing research infrastructure. Currently, already 2 services have been defined. So an ELSI, which stands for Ethic, Legal, and Societal Issues, Pediatric Services, based also on the previous TED experiences, will be able to ensure ethical, legal, and societal support and guidance for those conducting research with children, both biomedical and translational, from the preclinical phase to clinical studies, access policies, and education. And it will be specifically devoted to pediatrics and will be provided in collaboration with BBMRI, which is the already existing research infrastructure or biobanks. A second service will be a pediatric data interoperability service aimed to support use and reuse of data for research purposes, focused on discovery, access, integration, and analysis of biological data from a pediatric point of view. And this will be developed in collaboration with Elixir, which is the already existing research infrastructure on bioinformatics. So also in the future, EPTRI will aim to develop more collaboration within the ESFRI landscape according to the principles of strengthening the efforts while avoiding duplications. Where are we now? And I'm going to conclusion. I think I am in my 20 minutes time. The conclusion of the design phase has been marked by the submission of the conceptual design report to the European Commission. And after this, we followed also 2 further crucial steps that were the application to the ESFRI 2020 call submitted in May this year in order to get funding from the European Commission to support the preparatory phase of EPTRI, and the application to the ESFRI roadmap submitted last 9 of September to include EPTRI within the ESFRI roadmap and officially recognize it as a biomedical research infrastructure with a well-defined legal status. So now EPTRI is anyway approaching the preparatory phase, so the third step of the research infrastructure pathway to reach its legal, financial, and technical maturity, and finally the ERIC status, that is the European Research Infrastructure Consortium legal entity. Next two slides summarize the ESFRI project, which will encompass 3 different activities: networking activities, pilot transnational access to EPTRI research services, and finally joint research activities. And the last slide, and I'm going very fast to the conclusion, is the recently submitted application to the ESFRI roadmap. I just want to highlight that 16 governments supported EPTRI in this application, with other 2 governments also declaring interest for future evaluation. And we are working with even more countries, because this new research infrastructure will be really spread all over Europe and in many neighboring countries. So thanks a lot for participating to this webinar, and thank you for listening my presentation.
Thank you, Donato. Thank you for this really comprehensive overview on the EPTRI reality. Okay, so we can go now to the second speaker. I would like to introduce Professor Adriana Ceci, Gianni Benzi Foundation past president and CVBF scientific coordinator for EPTRI. Professor Ceci will talk about the pediatric drug development field and about challenges and opportunities in this area. Please, Professor Ceci, can you hear us?
Thank you, Angelica. Can you hear me?
Yes.
Okay, perfect. Thank you for your introduction. You can go to the first slide, please. Yes, in this speech, I would like to highlight the relevance of the biotech into the pediatric drug development as one of the main tools that we currently can adopt to implement pediatric medicine development. The second slide, please. Put the attention on the fact that as today, notwithstanding we have obtained many progress in the sense of increasing health of population, currently we still have a lack of medicine, for example, for some disease, like orphan disease, or for some population, and the pediatric population is one of the population in which the lack of medicine is still serious. Sometimes we also should take into account the fact that not every patient in the world have access to medicine that are already available. And this is mentioned as shortage of medicine. I mention this because currently we are dealing with the new proposed European pharmaceutical strategy that is exactly aimed to ensuring sufficient supply of safe and affordable medicine to every people. But in the context of this pharmaceutical strategy, it also includes the revision of the pediatric regulation and the orphan drug regulation. And this introduces a question. why children have the need to have a special legislation. Next slide, please. This is because, for example, just after the setting up of the European Medicine Agency, we can observe that the first line, the medicinal products for human use are constantly increased. And this increase in terms of medicinal products is quite parallel to the increase of active substances. That means that there are each year a number of new active substances, that means innovative medicine, that entered in the pharmaceutical market. The line on the back demonstrates that this increase is very, very lower when we speak about pediatric medicine and pediatric active substances. And what is really concerning is that, notwithstanding the approval of the pediatric regulation that has been at the end of 2006, the percentage, the proportion between pediatric and adult medicine is not really changed. That demonstrates that we already need to implement our efforts to change this situation. Next slide. This is the rate of approval of new drug in adult and in the pediatric, and you can see that regarding the new active substances, the rate of approval is lower respect to pediatric medicinal products. The means is that the gap is more visible in terms of innovative new pediatric medicinal products. Next one. This lack is evident in all pediatric age, but is extremely serious if you look to the number of medicine available for the very young children, like preterm, term, but also infant and toddler. And is only in the older children, adolescents in particular, that we can note that an increase of drug is visible. But this is not distributed in all the pediatric age. Next one. What is more relevant for us, as I anticipate, is the lack of pediatric medicine in the group where the need is more intense. For example, this lack affects the market of the orphan medicinal products, and in general, the market of innovative products like in advanced therapy or products from the biotechnology use in medicinal products. One point in this slide is of interest to analyze. Globally, we have in Europe 243 orphan medicinal products approved. Of these, a good percentage, 106 medicinal products, are approved for children. But another number, 127, is not approved for children, and is approved only for adults, even if in many cases the indication is of interest for the children. So we are not satisfied by the fact that in the orphan field, quite half of medicinal products are approved also for children. Because we know that the percentage of orphan disease that are of interest for children is more and more higher. So this is a gap that we have still to resolve. Next slide. So the pediatric regulation has been a big tentative to solve this limitation by introducing the concept that pediatric drugs must be developed in children differently than in adults because they need to use ad hoc methods and technology. And this is in general because the subset, the age subset have physiological characteristics that are different. And that these impact on all the pharmacotoxical, sorry, what happened? I have lost the...
Yeah, maybe there is some, please, for all the presenter, please mute your microphone.
Sorry, but I have lost my connection. Can you hear me?
We can hear you very well. Yes, yes, please. You can go.
Okay.
Ahead. Okay.
Okay, but really, I cannot see my my screen. But anyway, I can continue. Can you indicate me which is the next slide, please?
Yes, now we are showing the slide R&D in pediatric medicine limitation. So,
next slide.
Okay, challenges in drug discovery and development process.
Yes. This slide is demonstrating that after the arrival in the pharmaceutical developmental process of the innovative technology that are mainly linked to life science progress and data science progress, it has been a revolution in the pattern to develop drugs. And this has posed the problem that in the pediatric medicinal products, we should introduce this technology in order to maintain this market at the same level of adult pharmaceutical market. And you can see that are exactly the production of biotechnology drug represent one of the main challenges we have to deal with. Next slide is on
how to cover the pediatric medicine gap. Two main pillars, network and infrastructure.
Yes, I have completely lost my my connection. I cannot see my slide, and this represents a big problem now.
Yeah. Maybe you can try to open the slide on your computer. So I will.
My computer is dead. This is the problem. Okay. So I can have to change computer, and I re-challenge my presentation. You can go on with the next one. I'm sorry, but I cannot do anything.
Okay, maybe we can.
I am unable to solve because I don't have another computer home.
Maybe we can move forward, and maybe we can, in the final discussion, come back on this point and maybe.
Yes. Okay, okay. Okay,
you.
Angelica. I'm very sorry. Thank you.
Okay.
I can just try, Angelica, to connect by phone. If you give me just 1 minute.
Yes, please. In the meanwhile, I'm checking if there are questions. I remember you that you can type your question, also comment whenever you want in the question box. I will read them for you. Okay, I'm checking. No question at the moment.
Can you hear me?
Yes, Professor Cicci.
Yes.
Perfect.
Yes.
Okay.
I am connected with my phone.
Perfect.
So we can go on. Thank you.
Perfect. Okay.
So this is to say that faced with this problem, in Europe we have organized two main way to create a framework for resolving this gap. Networks have been largely used, for example, to organize clinical trials and clinical studies also in children. The second pillar is what Donato has explained before, the use of infrastructure that are more able to support in a stable way this kind of effort. Next one. So this is what EPTRI is aiming to do by the organization of his 4 thematic platforms. One of these thematic platforms is dedicated exactly to the implementation of biotechnology into the pediatric medicine developmental process. Next one. This is a picture of the Biosample Biomarker platform in which the research unit are mainly dedicated to the field of biomarker identification and validation, and also to give access to large and quality controlled biosample collection. We have a total of 23 countries participating in this platform for a total of 82 research units, and all dedicated to this field. Next one. Why EPTRI has decided to organize this kind of platform? The decision arises from the data collection during the survey and the discussion in the writing group of the EPTRI proposal. Because, as you can see here, in the group of need declared by researchers, the issue related to these activities have emerged in a very strong manner. There is the need to have a biobank. There is the need to study genetic factor linked to specific pediatric disease. There is the need overall to study polymorphism that impact also on biomarker development in the pediatric age.
So if we want to have access to the next phase of the innovation in medicine, that means to have access to personalized medicine also in children, we should implement this technology in our day-by-day capacity. Next one. So also during the survey emerged very well the interest of the research unit to a group of diseases that are very relevant for children. They regard the very early age of infancy, the rare genetic disease, but also neurological or respiratory disease. I means that there are a lot of relevant therapeutic areas that could be advantaged by the work that will be done in this platform. Next one. This is also to demonstrate that the relevance of this aspect has been tested during the previous EPTRI ID project conduct, and we have making evidence that there are two main needs expressed. One is to have access to well-annotated biological materials, both in terms of organized biobank and in terms of organized biosample and connected data collection. The second one is the identification of biomarker for pediatric disease, taking into account their variation during the pediatric age, and so to conduct validation study with reference exactly to this pediatric status. So we have confirmed the need to have a biosample and biomarker platform by using the data from EPTRI survey, the feasibility study, and we have translated this also in the next phase of EPTRI preparation phase. Next one. Those are the results for the survey. Demonstrate, for example, that in terms of biosample collection and biobank, EPTRI is able to offer the larger, until now, number of research units that collect pediatric sample and are willing to adopt quality and all the procedure that are needed for organizing biobank. Next one. This is possible because EPTRI is in contact with relevant clinical setting. This is done by the participation in EPTRI. We have mention of this in the user survey, for example, but also in the first phase of organization of the national EPTRI node, where many clinical settings are participating. So there will be a good collaboration, and this will give access to the pediatric sample. Next one. Here, the other area of research in the platform. I cannot spend time for this because we will have in the next of the today meeting other speaker that will explain which will be the excellence of this kind of research. Next one. as I said, we are confirmed the need to have these activities in EPTRI, because during the feasibility phase of EPTRI, we have collected the proposal of studies by many users, and we have demonstrated that on a total of 10 feasibility studies proposed to EPTRI, many of them were related to the need to have activities that are located into the biomarker platform, to have identification and validation of biomarker, omics application, biobanking access, IT tool to use data associated to biosample. Next one. Finally, as I said, we will have, we hope, very soon, the opportunity to test this capacity of EPTRI, because in the ESFRI project, it has been included the proposal of a joint research activities that is aimed to demonstrate the capacity to put together this large scientific community and to start to provide this kind of service to the whole pediatric community in Europe. Thank you.
Thank you, Professor Ceci. Really clear, comprehensive overview on the framework behind EPTRI, the challenges in the pediatric drug development field. I will move forward now directly to the second session, since we are slightly late, and we will have a question and answer session at the end of the innovative approaches in pediatric drug development session. So I would like to present the first speaker of the second session, Pierluigi Martelli from University of Bologna, a national contact person for the interoperability platform at Italian node of the ELIXIR, the European Infrastructure for Life Science Information. Pierluigi will talk to us about omic application in drug target discovery and preclinical research.
Thank you. Can you hear me?
Yes, and
I share the screen.
Yes, perfect. So the floor is yours.
Okay, thank you very much. Thank you for the kind invitation. I'm Pierluigi Martelli. I'm a bioinformatician at the University of Bologna. I work also in collaboration with ELIXIR, as mentioned, and I will introduce quite briefly what can be the applications in the field of drug target discovery and in general in the preclinical research of the omics techniques. We are living now in the so-called omic era. The omic era starts conventionally in 2001, when the grafts of the complete genome of humans were released into two famous issues of Nature and Science. And omic means the ability of investigating the components of a living organism or of a tissue or of a cell in its entirety. And from 2001, we have developed several techniques, biotechnological techniques that allow to investigate the different molecules that are present in a living system in different aspects. So we can have information about the genome of an organism, about the variations that are present in a population. We can have interpretation of the genome section, the genes, if you want. We can have the techniques for investigating the proteome and the epigenome and so on. on. And this is very relevant because these techniques, of course, are a very powerful tool to address what is the holy grail in molecular biology, that is this section of the relation between genotype and phenotype. Very roughly, and if you want, very naively, we can say that DNA contains information that we call genes in general. So the human DNA that consists of 3 billion bases, 3 billion letters encodes for 20,000 protein coding genes that overall have the instruction to build some 40,000 proteins and many other RNA transcripts. And these molecules are the machinery that perform a function in the cells, and the result of this function is the phenotype, so the traits that characterize each individual. And of course, this relation is very important in the case of pathologies, because even this very naive view was conducive to large discoveries. The fact that, for example, many mutations, many variations on the genome can end up in variation, for example, on proteins that at the end hampers the machinery and give rise to disease or hampered phenotypes. And of course, the availability of techniques, of genomic techniques, in particular of DNA sequencing, that allow to obtain in a short time and in a costless way the complete genome of an organism, or the complete genome of a tissue, if needed in the case of cancer, for example, in a very short time with a technique that I cannot explain, but is based on the fragmentation deep fragmentation of the DNA and the sequencing of the short fragments that are then assembled with computational techniques. These allow to obtain now a genome, in whole genome, with a cost of around $1,000. And in 20 years, the cost of the genome sequencing lowered by 5 orders of magnitude, so an enormous increase. And these, of course, allow to perform larger studies that involve the sequencing of populations, and, for example, allow to conduct large case control studies or quite large pedigree studies, in which we can discover what are the significant variations that differentiate people that carry a particular phenotype, in particular that carry a disease, from the DNA of people that can be considered as healthy. And this work is not easy, because you have to consider that as a baseline, between two random individuals, around 4 million sites are different. But if we have a quite large population, we can end up with a set of variations that possibly target some molecule, possibly target some protein, possibly are influencing the expression or the structure of some protein. And this, of course, will give the possibility of choosing drug targets and to check if some library, if some with some drug library, if there are possible therapies to be discovered. Of course, this process is difficult, because even in a large case study, case control study, or in a large pedigree analysis, we end up with tens or hundreds variations that are possibly implicated in the disease insurgence. And this is too much. We need computational methods for prioritize these variations. And in particular, we have to leverage the information that is contained in the current databases. And so to date, we have a quite good association among some 30,000 variations and and diseases, variations that happens to occur in 13,000 human proteins. So we have this known association between variation and diseases, and we can use the so-called machine learning frameworks, so artificial intelligence, if you want, or methods like deep learning, to understand what are the rules, what are the features that characterize these variations. So to use the available information to extract general rules that are encoded in machine learning methods, and then to apply these rules to new variation in order to score them and to score the likelihood that a new variation is related to the disease. This is a big issue, and in Bologna, we developed different tools to this aim. I will briefly present in this slide two of them. Snips and Go. Snips and Go is a tool that taken a sequence, taken a protein sequence and a variation of the protein sequence, is able to predict whether the variation is related or not to a disease. So it gives a score for the variation to be pathogenic, if you want. And Snips and Go, when compared with other methods, well performs. This is more or less the state of the art. Nowadays, we have methods that can predict correctly, can give the correct prediction for about 80–85% of the variations. That is not a perfect prediction, but of course this can help in the prioritizing drug targets. On the same way, we developed also INPS, that is a method to predict whether a variation on a protein sequence or on a protein structure can lead to a change in the free energy of folding of the protein. So we can predict whether a variation can stabilize or destabilize a protein. And also in this case, the prediction is not perfect, but we have a correlation coefficient between the delta G predicted and the real delta G, that is 0.7. That is not too bad. These tools can help the finding of new targets, so to find proteins that can be implicated in the surgence of the disease. But of course, even if this naive view, gene phenotype, gene protein, so gene mutation, protein mutation disease is very simplistic, this view has been very successful in some diseases, but this view is too simple because the relation between genotype and phenotype is much more complex. So we know that genes encode for proteins and proteins correspond to function, but proteins are not always present in a cell, and not all the proteins are present in all the cells because the cells are different. So proteins or genes need to be expressed, and the level of investigation of expression is very important. And the mechanism of the expression involves, for example, the interaction between proteins and DNA. And proteins, when they are expressed, protein interact. They form a network of interactions that are very specific. Pairs of proteins interact in a specific way, and these interactions are at the end conducive to the emergence of metabolic pathway, the fact that different proteins and different molecules in general cooperate in a sequential way with a very complex diagram to perform functions. So different proteins cooperate to perform a complex function. And at the end, the effect of variations, the genetic imprinting or the genetic information is to be analyzed in this complexity. And for that reason, we need omic data. And it is very nice the fact that the biologists were able to use a sequencing technique to expand this technique in order not only to analyze the genome of an organism, but also to analyze, for example, what is the transcriptome, what is the level of messenger RNA, so to understand when and where genes are expressed, but also to understand which are the possible interactions between proteins, transcription factors, and DNA, and also to understand what is the state of the chromatin, so the state of the complex between DNA and proteins that is essential for gene regulation. And going further, we can apply also sequencing techniques, so the same techniques that I briefly introduced, also for investigating communities of organisms like the microbiomes, like the gut microbiomes, but also the microbiome that live in our environment. Not only sequencing is required, because different levels of omic can be investigated, have to be investigated to achieve a complete knowledge of the biological system. And in particular, we need, and we have, techniques for exploring the proteome, that is to understand when and where the proteins are expressed at the level of the tissues, of the human tissue, but also at the level of the cell, where the proteins is localized at the level of the cell, with mass spectrometry, with immunofluorescence, and so on. We can also have, and we have to apply techniques to understand how the proteins interact. What is the pattern of interaction between different proteins? We have different techniques like striped and mass spectrometry again, but beside proteins and besides nucleic acid, we have also to explore what are the metabolome, what is the concentration of the small molecules inside the different tissue and districts. And for that reason, we have the possibility to use metabolomic techniques. So all this level of investigation investigated different species of the complexity of the biological system, the genome, the transcriptome, the proteome, the metabolome, and in each level we can understand what are the interactions, so we can understand what are the interactions between proteins. And we have to connect all this data, we have to integrate this data. Of course, this data needs, first of all, to be stored and organized into databases, and then to organize in different conceptual framework like pathways, networks, and then they need to be integrated because only with the integration of the complex system we will be able to discover new targets, because sometimes a protein is mutated, but the correct target is a protein that is interacting with the mutated protein, or the protein is mutated, but the effect at the end is an effect on the expression on DNA, so we can search for other targets. But also this complexity of interaction give rise, for example, to side effects, because we are targeting a protein with a specific drug that can solve a problem, if you want, but gives problems because of the complex interactions in which the targeted protein is inserted. So we need more and more computational methods. And to the same, for example, in Bologna, we developed an integrated database called EDGAR that takes some not all, but a large data set of known gene disease associations. We are expanding, we are releasing now in a few months a new version, but the important idea, in my opinion, is that we insert each of the gene that is causative to disease or associated to a disease in the context of its interactions. So we are analyzing what are the interactions that are reported in different databases for the protein encoded by the gene, what are the known interactions that are superseding the regulation of the gene, and we are investigating the gene in the context of the pathway and the functions that are characterizing the gene. And so for each gene, for example, we can understand what are the interaction, the regulatory relations, the functional relations, and for example, if you have two proteins that are involved in a particular disease, because sometimes different proteins or different mutations end up in the same phenotype, then we can understand what are the common partners of interaction. And these common partners can be a new target for the therapies. We can understand if the proteins involved in the similar disease or in the same disease are co-regulated, share some transcription factor, and again this transcription factor can be maybe a new target, and also understanding what are the pathways, what are the functions in which the protein, the mutated proteins live, of course, can expand the possibility to find targets that are related to the function that maybe is hampered by the mutation. Moreover, we added a layer that involves the phenotype, so we can computationally exploiting the information that is present in the database, we can connect each phenotype to a set of disease or to a set of genes and to a set of mechanisms that can be involved in the phenotype. And for example, we can take three phenotypes that are maybe present in some patient. This is just an example. And for these three phenotypes, we can understand what are the possibly affected pathways or the possibly involved pathways, and what are the possible genes that are influencing the three phenotypes. And so we can have a picture of the genetic basis and of the functional basis that are at the basis of a co-presence of the co-occurrence of phenotypes, and this can lead to the personalized medicine in order to identify for each group of patients with a particular set of phenotypes what are the possible targets for the interventions. In conclusion, the omic techniques provide very powerful tools to dissect the complexity of biological systems, and more the drug targets and the drug effects and also the side effects have to be explored in the context of this complexity. But to do that, we need the data to be collected, of course, experimentally collected, stored, biosamples have to be stored, but the data have to be collected, standardized, integrated, and analyzed. And in this context, I would like to present very briefly ELIXIR, that is a European research infrastructure that is starting collaboration with EPTRI. It is a landmark European research infrastructure that collects 23 governments, so 23 different nations in Europe, and is coordinated by one hub that is located in Kingston in the UK. And ELIXIR has the scope, the goal to bring together bioinformatic resources for managing data, for managing tools for analysis, and to provide training, and also to provide the computational power to perform computations, to perform all the analysis that are needed in different fields, including, of course, medicine. With this, I thank you. This is the website of our group in Bologna. If you are interested in more details, you can contact me in any time. Thank you for the attention.
Thank you, Pierluigi. A really nice presentation describing tools available for the drug target discovery. Thank you so much.
Thank you very much.
We can move on the second presentation. I just remind that you can type your question for the audience, type question in the dedicated box, and I kindly ask all the presenters to be on time, so not to exceed the 20 minutes for the presentation. The next speaker, Emanuel Nikros from Athena Research Innovation Center in Information, Communication and Knowledge Technologies, is the coordinator of the EPTRI platform dealing with pediatric medicine discovery, and he will give us a presentation about a novel preclinical model as platform for drug discovery for pediatric diseases. Please, Emanuel.
Hello, I hope that you can see my screen. I'm not sure.
Yeah, we can see your screen.
Okay. Is the GoToWebinar also, you can see also this part of GoToWebinar? Okay, doesn't matter. Thank you very much, Angelica. Since a century ago, Paul Ehrlich set the basis of the drug discovery process by introducing the concept of the receptors, so the magic bullets that are molecules targeting specific receptors, and the process comprising iterative steps of chemical synthesis and pharmacological evaluation. More or less, we have the same concept actually a century later. The last 40 years, well-known and specific milestones have been established in drug discovery. They can be summarized by dividing them in the preclinical part and the clinical trials. We all know that it is a long process and a quite expensive one. In 2016, the cost has been estimated to be close to 3 billion to bring one molecule in the market. The first phase in the drug discovery, called usually R&D, consists also of specific steps that are related with target validation, hit identification, hit-to-lead optimization. Several newly developed methods have been introduced to accelerate the process. We've heard in the previous talk, omics for target identification, high-throughput screening for hit identification, and also we've heard about all different in silico modeling and that can be used in rational drug design, among others. From lead to candidate, the steps are related to animal studies. Efficacy and toxicity are the most important. However, administration, formulation, quality control, etc., are also issues to be solved at this phase. The human proteome, you've heard about it, contains approximately 20,000 proteins, I'm sorry, and many of them are potential drug targets. At the moment, drugs are known for about 700 among them. It is interesting that the number of known targets for approved drugs has been doubled within 10 years after the human genome sequence deciphering. Bioinformatics and statistics show that certain target families like GPCRs or nuclear receptors have shown to provide more safe choices for drug discovery. In this slide, when in a target family, the orange bar is higher than the blue one, the target is more likely to prove effective and eventually deliver a drug in the clinical use than the other target families where the blue bar is higher than the orange one. This situation creates a tendency to focus more on successful targets while neglecting others, and this applies mainly in rare diseases, and pediatric diseases are often rare. Actually, it is expected that the druggable targets will increase from 700 for small molecules and 150 for biologics up to around 5,000. However, the druggability of a target, which is the possibility to modulate the target function within the context of a whole organism to treat a disease using a synthetic compound, is usually very difficult to be validated. Very often, the druggability of a target might only become apparent in retrospect once the program has advanced considerably, or if drug-like molecules are already known to exist for the specific target. Coming to biotechnology and techniques that are important for drug discovery, it is the well-established biotechnological methods of great importance in drug discovery are the overexpression and purification of proteins. Those proteins will be used in high-throughput screening campaigns, and all big pharmas have set up robot systems to accelerate these processes. The HTS is based very often on different biophysical and biochemical methods like SPR, DSF, FRET, MST, ITC, to name the most frequently used. And screening method, when the screening method results to important details on the protein ligand interaction, usually is more demanding and less high throughput, notably X-ray crystallography or NMR. And in that case, only a few hundreds of molecules can be studied, while in other methods can screen up to millions of molecules. That brings us to the chemical libraries issue. As I have shown previously, the biological relevant space consists of a few thousands of protein targets. The drug-like chemical space is tremendously larger. It counts about 10 to the 60th, and this is for drug-like molecules. It's larger than the number of stars in the universe. The actually known chemical space is about 100 million 10 to the 8 compounds. In order to track hit molecular scaffolds, the drug discovery campaign nowadays begins with high-throughput screening of a large chemical library. Thus, all big pharmas have organized quite important and completely robotized libraries comprising millions of molecules. In the next step, the main aim of the medicinal chemist is to synthesize molecules based on the hit scaffold with high affinity to the target, reasonable selectivity, and adequate ADMET properties, so that they may hopefully produce a desirable therapeutic effect. In order to underline the effort of this initial phase, I also show in this slide that it is generally accepted that in order to have a final drug in therapeutics, at least 10,000 should be synthesized and tested. Take-home message: without chemical synthesis, new chemical synthesis, there is no drug discovery. Coming back to biotechnology, useful technologies, novel technologies that are important to mention have been developed the last 10-15 years and are very important for target validation and understanding mechanisms of disease are gene editing by CRISPR-Cas9 and the use of siRNAs to downregulate the expression of specific proteins. The other important part of drug discovery, apart the chemistry one, is the animals. Is the other important component of drug discovery, and the generation of animal models for each specific disease is of utmost importance. Transgenic mice are well established. However, for several diseases, it's difficult to produce such models. Another important animal model is zebrafish, has proved to be a very useful one. It is optical transparent, both embryos and adults, and enable direct observation of cell behavior. The other advantage is the rapid production of zebrafish animals, and finally the significant conservation of signaling pathways between fish and humans. In this slide, I summarize more recently emerging technologies important for cell-based assays. The most important are the pluripotent stem cells and 3D organoids, microfluidics, the gene editing that I have already mentioned, and new developments of cell imaging. The in vitro modeling of human diseases was revolutionized by the discovery of pluripotent stem cells by Takahashi and Yamanaka in 2006. Patient-derived iPSCs offer an invaluable alternative for modeling rare diseases. By utilizing these pluripotent capacity stem cells that carry specific pathogenic mutations, we can have patient-specific cells and model the molecular mechanisms underlying disease pathophysiology. In this slide, I refer to two papers describing the importance of using this kind of stem cells. It is related to autism spectrum disorders. It has been successfully modeled using iPSCs, pluripotent stem cells. We know that autism are characterized by impaired social interactions and communication deficits. Animal models cannot be used in autism modeling. In the specific two papers, we have in the first paper, the authors based on the observation that cumulative gene analysis in ASD subjects, stem cells of ASD subjects, have identified several mutations in a specific protein, a specific gene that express the SANK3 protein. This regulation of SANK proteins lead to the alteration of NMDA and AMPA receptors trafficking. They have shown that treatment of neurons that have been differentiated from stem cells from patients promotes the formation of mature excitatory synapses by increasing AMPA and NMDA receptors. And thus, in the second paper, we have a double-blind placebo-controlled phase 2 trial where is described the using of IGF-1 treatment that resulted to significant improved social impairment and restrictive behavior of the patient. The other important development are the 3D organoids. It is well known that 2D monolayer cell culture have been in the drug discovery pipeline for several decades. They are less useful in modeling diseases where cellular microenvironment plays a vital role. For years, scientists have been working to develop 3D culture systems. Organoids are typically derived from different types: adult stem cells, embryonic stem cells, pluripotent stem cells. And we can think of them as miniature versions of organs. They often display a very accurate microanatomy and have been established for many organs like lung, brain, liver, stomach, kidney, etc. And in the slide, you can see an example of a paper describing organoid models of childhood kidney tumors. Finally, based on these technologies, organs on a chip and even multi-organ chips can be derived using microfluidics. In the next slides, I will also describe a representative study proposing how the whole concept of target validation by the use of sophisticated biological tools can be applied to pediatrics. It concerns pediatric glioblastoma. There is very few options currently available and very poor results to treat this disease. So discovery of novel pharmacological targets are of primary importance. In the study, the authors propose a stepwise approach consisting of the sequencing of patient genome, transcriptomics, target validation, and translation to the clinical practice. In the first step, the whole genome sequencing effort was implemented. It resulted to a number of critical genes that was impaired, and many of them were involved in pathways of key importance. Within these, several protein kinases were identified. In the second step, the transcriptomic profile of the disease was determined. The most interesting result was that the gene encoding MET kinase, which is a well-known oncogene, was the most frequently affected gene, and this gene was found to form fusion with other gene products. So it was considered as a promising target. The next step it was to use small molecule kinase inhibitors, and the researchers began studying in vitro cells and specific carrying pathological MET fusion products. Then they continue to allografts and to xenograft models in mice. In all cases, the administration of MET inhibitors showed a clear improvement of the phenotype. The most interesting result was the significant prolongation of survival of the group of mice that were treated with forretinib. So these encouraging results led the researchers to consider the use of MET inhibitors as a therapeutic strategy against glioblastoma. So they use it in a specific pediatric case. So the juvenile patient was administered the drug crizotinib, and a marked response was monitored in the beginning of the study. Unfortunately, resistance then emerged. So overall, the specific example shows the potential of these methods in understanding mechanisms, personalized treatment, and rational targeting validation. Under the light of all the previous, EPTRI has focused on the gaps and the needs of the pediatric disease drug discovery and organized a platform aiming to offer access to academic drug discovery facilities specialized to pediatric diseases, provide integrated services by coordinating the European Centers of Excellence, and implement cutting-edge technologies in order to accelerate discovery in pediatrics. In this slide, we try to gather all different technologies needed in preclinical phase of drug discovery. You can see 6 different groups of technologies and methods. Based on a survey gathering responses by scientists from all over Europe, we identified gaps in discovery efforts pursuing pediatric drug development. Those gaps demonstrate the necessity for a dedicated and original research platform in the European Union. The most important are understanding mechanisms and differences between children and adult pathologies, and establish targets, new targets for pediatric diseases. In the same context, most existing disease models are inadequate to capture the unique developmental status of pediatric patients, whether we are talking about cell models or animal models, and thus specific disease models are needed. And finally, we believe that the personalized medication approaches in pediatrics are not widely exploited at present. The last 2 years, EPTRI efforts pulled together information in European level for a unique collection of disease-specific cell models. You can see muscular dystrophies, cancer, neurological, etc. And the use of all the different state-of-the-art technologies that I have described before for 3D organoids, stem cells, etc. However, the most important are the existing animal models. You can see in the slides, models from Drosophila fly up to mice through zebrafish for different kind of diseases, specifically pediatric diseases. And thus, in this context, the EPTRI platform for pediatric drug discovery aims to provide services in target validation, characterization, providing chemical probes, bioinformatic services, or genetic approaches like knockout, knockdown, or silencing genes, etc. Cell models, all the previous described technologies, stem cells, organoids, microfluidics, gene editing, etc. Animal models, you can see in the slide more animal models in the collection, zebrafish, rabbits, rodents, for always in the case of different pediatric diseases, even mini pigs, mainly for pharmacokinetics, and apart the drug discovery itself, meaning the assays on new molecular entities. It is important for the understanding of mechanisms of toxicity, of drug metabolism, and even pharmacokinetics, although it is in a further discovered from another platform of EPTRI. Finally, all the different kind of in silico studies for virtual screening, for heat to lead design, help medicinal chemists for prediction of ADME properties, of toxicity, etc. There are more than 100 centers of excellence in whole Europe that are able to provide all these services. Finally, EPTRI aspires to develop a new structure where government, academia, industry, and citizens work hand in hand by combining basic and applied research over a multitude of interdisciplinary fields spanning from bioinformatics and developmental biology to advanced animal models. EPTRI drug discovery platform, we hope that will facilitate and accelerate drug development, focusing directly on pediatric populations. And with that, I would like to thank you.
Thank you, Emmanuel. Thank you for this really comprehensive overview on the available models useful in the preclinical research for the pediatric drug discovery and development. Next, we will have the question and answer section at the end of this session, so we can move on the third speaker. The next speaker, Marios Philaktides from Cyprus Institute of Neurology and Genetics, that is the coordinating institution of the EPTRI platform dealing with the pediatric biomarkers and biosamples. Marios will talk us about biomarkers and personalized medicine in pediatrics. Please, Marios, the floor is yours.
Hi, hello, can you hear me okay?
Yes, perfectly.
Okay, I was going to say every 20 minutes there is a small break in the sound for about 20 to 30 seconds. So if that happens during my presentation, let me know and I'll go back over the points that were missed.
Okay.
So today I'm gonna give a general overview of some of the problems of using biomarkers in personalized medicine in pediatrics, and then talk about asthma as a condition where biomarkers are utilized for achieving a more personalized approach to treatment. Okay. Okay. So prior to the 1970s, children were essentially excluded from clinical trials from new drugs due to the perceived ethical challenges and difficulties in studying this patient population. And exclusion from clinical trials resulted in a deficit of proven and effective treatments for children. Later on, in around 1977, the American Academy of Pediatrics Committee on Drugs published a statement saying that it was unethical to force physicians to prescribe medicines to children without adequate information on drug dosing and safety generated in this population. The committee said that it was critical to conduct appropriate drug studies in children to establish safety and efficacy, especially when children were the intended population for the drugs. Although today the pediatric therapeutics knowledge gap has been narrowed, the current knowledge is still inadequate in certain areas to support pediatric drug development at the same pace and quality as for adults. And unfortunately, the implementation of new research technology and findings for children still typically occurs well after investigations have been completed and used successfully in adults. Additionally, the same rigor of research is often lacking when testing or validating new technologies and innovations in children due to the perceived and sometimes real barriers in pediatric research. Two major areas of need in pediatric drug research are developmental disease characterization and developmental pharmacodynamics, which are currently limited by lack of appropriate biomarkers. Yes, so personalized, so did I say, yes, sorry. Personalized or precision medicine have become very popular concepts both for adult and children patients. Many different definitions exist for these terms, which are sometimes used interchangeably. But precision medicine, as defined by the Precision Medicine Initiative, is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. Now, the simplest definition of personalized medicine comes from the Pharmaceutical Committee of the European Commission, and it says that personalized medicine is the provision of the right treatment to the right patient at the right dose and at the right time. Also, the concept of stratified medicine also exists, which is the selection of the best approach to managing a group of patients. So this is a more broad concept than personalized or precision medicine. The precision medicine research can be categorized into two broad goals: precision diagnosis and precision therapeutics. Precision diagnosis involves understanding and characterizing the biochemical processes that have been perturbed, which result in pathology. Additionally, precision diagnosis research aims to uncover genetic and environmental contributions to disease and long-term prognosis. The second goal of precision medicine, that is precision therapeutics, is to determine the correct therapeutic agent and the dose that achieves the desired response while anticipating and mitigating adverse reactions and events. Flow of information between precision diagnosis research and precision therapeutics research is critical for the advancement of precision medicine as a whole. So in order to achieve the goal of precision medicine, both precision diagnosis and precision therapeutics are required, and biomarkers are the key to achieving both of these. So a biomarker, the FDA defines a biomarker as a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. So as can be seen from this figure, biomarkers fall into many different categories. Some of the most commonly used categories of biomarkers include diagnostic biomarkers, which are used to identify individuals with a disease condition of interest or to define a subset of the disease. Prognostic biomarkers are also very common, and they're used to identify the likelihood of a clinical event, disease recurrence or progression, and predictive biomarkers, which are used to identify individuals who are more likely than similar patients without the biomarker to experience a favorable or unfavorable effect from a specific intervention exposure. Typically, biomarkers developed for use in adults are not extensively validated in children. An additional issue with pediatric biomarkers is often the lack of clear informative values within the pediatric population. This can limit the utility of a biomarker, since an ideal biomarker value that is associated with positive health outcomes has not been established. One of the problems of delivering precision care for children is the fact that the characterization of pediatric disease can often be incomplete. And the reason for inadequate characterization is partly because pediatric diseases and conditions are often interpreted through established adult symptomology. For example, in juvenile rheumatoid arthritis, was initially assumed to be similar to the adult rheumatoid arthritis. The disease in children was assumed to have the same underlying pathophysiology and therefore the same prognosis as in adults. However, it was later established that juvenile rheumatoid arthritis is far more heterogeneous than the adult onset rheumatoid arthritis. So with a pathology that has a lot, much higher heterogeneity, part of the challenge of caring for the pediatric patients is to accurately determine the disease subtype and the appropriate intervention for this specific subtype. Biomarkers used to monitor adult onset rheumatoid arthritis are also inadequate for describing the juvenile version of the disease, because only a small percentage of the pediatric patients also have a form of arthritis which resembles the adult disease. So again, this example highlights the fact that pediatric-specific biomarkers are clinically important for accurate diagnosis and pediatric care. Understanding the impact of developmental stage on pharmacodynamics responses is critical in designing therapies and developing drug regimens that can achieve precision medicine for children. Improved knowledge in pediatric pharmacodynamics needs to complement the current understanding of how developmental changes affect pharmacokinetics in children. Both pharmacokinetics and pharmacodynamics of a given drug may be highly variable in a developing child, particularly
Marios, can you hear us? We cannot hear you anymore. Marios, can you hear us?
Helping achieve personalized medicine.
Marios, we lost you on the previous slide.
Oh, we lost me on the previous slide. Okay, yeah. So let me just go over the notes there. Sorry.
Yeah. Okay.
So what I said there was that understanding the impact of developmental stage on pharmacodynamics responses is important in designing therapies and developing drug regimens that can allow for precision medicine in children. And improved knowledge in pediatric pharmacodynamics needs to be combined with the current understanding of how developmental changes affect pharmacokinetics in children. So both pharmacokinetics and pharmacodynamics for a given drug can be very variable in developing children, and both must be taken into account when making decisions about the doses of drugs in children. And the concentration of drugs in children can vary between children of different ages, between children and adults, because the expression of the drug target can be very different between adults and children. So looking at asthma as an example of a disease where biomarkers can aid in the provision of personalized care. Asthma is a heterogeneous and multifactorial respiratory disease with an important impact on childhood. Childhood asthma is a common chronic airway disease characterized by airway inflammation, airway hyperresponsiveness, and reversible airway obstruction. It affects about 15% of school-age children in Europe and has an increasing incidence and prevalence. The symptoms include wheezing, shortness of breath, chest tightness, and cough, ranging in severity from mild symptoms to life-threatening exacerbations. There are international guidelines for the management of pediatric asthma based on clinical factors, and these have been established in order to control the symptoms and to reduce the risk of future associated complications. And pharmacological treatment has a key role in achieving asthma control. Reliever medication aims to alleviate the symptoms during the worsening episodes of the disease, with short-acting beta agonist or SABAs being one of the most common reliever medications in children. And when SABA monotherapy is insufficient to prevent symptoms, controller medication is recommended. The most commonly used controller medications are inhaled corticosteroids. And inhaled corticosteroids have an anti-inflammatory and immunosuppressive effect on lung tissue. As asthma severity increases, inhaled corticosteroid dose may be increased or combined with other controller therapies such as long-acting beta agonists or leukotriene receptor antagonists. Children with severe asthma require add-on therapies such as oral corticosteroids, which are similar to the inhaled corticosteroids, except for the systemic effects derived from oral administration. However, the incidence of adverse reactions increases with oral corticosteroids, and this limits their application to the most severe cases. So, despite following asthma management guidelines, some patients still have sustained symptoms under treatment with high-dose inhaled and oral corticosteroids. So, personalized medicine has recently emerged, aiming to select the most appropriate therapy for each patient based on identifying different asthma phenotypes and endotypes. Childhood asthma phenotypes differ in clinical presentation, natural history, inflammatory mechanisms, response to treatment, and also depend on age. The increasing awareness of heterogeneity of childhood asthma led to the recognition of different asthma endotypes. And an endotype is a subtype of a condition defined by a unique or distinctive functional or pathophysiologic mechanism. There are two major endotypes defined for asthma: type 2 or T2 high asthma and type 2 low asthma. And these are based on the type of the underlying airway inflammation. Now, type 2 high asthma is typically characterized by eosinophilic inflammation initiated by alarmins such as interleukin-25 and 33, which are secreted after a trigger in the bronchial epithelium. And this trigger could be allergens, microbes, or pollutants. And subsequently, the levels of these interleukins are sustained by the release of signature cytokines such as IL-4, IL-5, and 13 from cells of both the innate and the adaptive immune systems. These type 2 cytokines actively then recruit eosinophils, mast cells, and basophils in the airways and directly mediate IgE synthesis and contribute to the hallmarks of asthma pathophysiology. Type 2 high asthma endotype typically displays a good response to corticosteroid therapy and has become the target of biological therapies. Now, type 2 low asthma is less known and less studied, and it is driven either by neutrophilic or less commonly a granulocyte poor inflammation pattern sustained by IL-8, 17, 22, and other T-cell related cytokines and epithelial cell-derived cytokines. T2 low asthma endotype is rather rare and is mainly seen in patients with severe disease and shows corticosteroid insensitivity. So this table, we see some of the most important biomarkers used for the management of pediatric asthma. Biomarkers for asthma can be measured in different biological specimens, including sputum, bronchoalveolar lavage, exhaled breath condensate, bronchial biopsy, urine, and blood. At the moment, bronchoalveolar lavage and with bronchoscopy and bronchial biopsy are the gold standard to assess airway inflammation and remodeling in asthma. However, the invasiveness of these diagnostic methods limits their use in children in daily clinical practice. Sputum induction still has limited use outside the research setting, as it is considered semi-invasive, as well as technically complex, especially in children younger than 8 years of age. Thus, the availability of non-invasive methods to study and monitor disease inflammation is of great relevance in childhood asthma. These biomarkers shown here for asthma have potential utility to distinguish the inflammatory endotype T2 high versus T2 low asthma, and predict responsiveness to specific treatments, in particular T2 cytokine targeted therapy, monitor success for a selected treatment option, and assess the risk of disease progression. Single and combination biomarkers are now being recommended for use in the assessment of patients with asthma, and in particular with severe asthma. Most of the current established biomarkers, as can be seen in this table, are available in clinical practice, are related to T2 high inflammation. And these include blood or sputum eosinophils, serum IgE, serum periostin, and fractional exhaled nitric oxide. Of course, there are many more candidate biomarkers which are under investigation, and omics technologies are uncovering new candidate biomarkers such as SNPs and microRNAs and metabolites.
Marios, can you hear me? Marios, we lost you again.
Monoclonal antibodies against either IgE. Yeah.
Marios, we lost you again.
At
the end of the slide, actually. So
at the end of the slide, okay. So
of the previous slide, yes.
At the end of the previous slide. So what I was saying that in addition to these biomarkers, which are shown here, which are mainly phenotypic biomarkers, a lot more candidate biomarkers are under investigation that are uncovered by omics technologies, such as SNPs, metabolites, microRNAs, methylation patterns. However, these are still at very early stages of research, and these have not made it to the clinic. That's why I'm not going into any of those here. So in this slide, what we have is the like I was saying, the flip side of the coin, where we're looking at biologics for the treatment of asthma and the predictors of their response. These biologics are generally humanized monoclonal antibodies against immunoglobulin E, IL-5, 4, and 13, and have been approved by the FDA for use in children as young as 6 years old. And for each of these biologics, there are predictors of response. Some of those predictors are the biomarkers that you have seen in the previous slide. So for omalizumab, for example, elevated fractional exhaled nitric oxide, elevated serum neutrophil are some predictors of response, together with high body mass index of over 25 and exacerbations of the disease. Just wanted to list some of the publications which were very useful in having a look in preparing this presentation. So if you want to find out more, you can look at these. So thank you very much, and that's it from me. Thank you.
Thank you, Marios. A really clear introduction on how biomarkers can be really useful in personalized medicine for children. Thank you. The next speaker, I remind you that you can type your question or comment in the dedicated box. And now I move on the next speaker, Giovanni Migliaccio, Scientific Director at CBF. that will be giving us a presentation about pediatric application of ATMP. Okay, you can share your screen.
Good morning.
Okay.
Dear ladies and gentlemen, I will try to be concise as much as I can. What are the advanced therapy medicinal products? Well, the term has been introduced in Europe by a legislation. It's the regulation 1394/2007 that introduced the terminology and the definition of advanced therapy medicinal product, and also changed two other pieces of legislation, the directive on the marketing authorization for medicinals, and the regulation that institute and describe the functions of the European Medicine Agency. So what kind of things are the advanced medicinal product, for short ATMP? Well, there are three categories: the gene therapy medicinal product, the somatic cell therapy medicinal product, and these two categories were not new. They were already described in the previous Directive 2001/83 in the Annex, in Part 4 of the Annex 1. So this was material that was considered a medicinal product already in 2007. 2001. But they added a third category, the tissue engineered products. These are kind of a mixing between cell, gene, and something. It can be a medical device, can be a structural base, can be an engineered material, a lot of different things. So it's been some kind of strange piece of legislation, because it was introduced not so much on the need, but more on the prevision or forecasting of needs. So after a while, the Commission asked a report on the application of ATMP in 2014, and concluded that the regulation has protected patients from unsound treatments. However, it also recognized a few shortcomings and identified actions needed to adjust the piece of legislation. What kind of unsound treatments? Well, if you had a look to newspaper and news, you would find advertisement for what is called regenerative medicine, for stem cells. A classic was a goat stem cells for rejuvenating, which is kind of interesting. But there were many that was much more dangerous than this. So the fact that the regulation was in place was effectively in protecting the European citizens. Another action was taken by the European Medicine Agency, holding a multi-stakeholder meeting in 2016. In that meeting, there was industry, representative of the patients, physician, the members of the specific committee or ATMP, the CAT at EMA. So it was kind of discussing what was going on, which kind of product were under development, what were the obstacles at a faster development. But in all of this, in the report, there was no mention of pediatric patients. The focus was on regenerative medicine, which is a terminology that is appeared in the EU lexicon in the regulation for the tissue engineering product. Now, what is changed in this year? Well, the first thing is that we have a number of products licensed. So 14 different products were licensed under this regulation by the EMA, which is still a small number in comparison with the classical or biological product. Moreover, of these 14, these 5 were retired from the market from different regions, maybe just for a low return on investment, for high cost, and so problems in reimbursement. So all of them were retired, not retired by the agency, but retired by the producer that found uneconomic to keep them there. These 5 instead are the new generation. Here you can see that these products are targeting, well, cancer and some other kind of degenerative disease like holoclar, that is destroying a missing stem cell for the repair of the human corneal epithelia. But the new one are for CAR-T application. And this is the new wave of products coming through the pipeline as the use of genetically engineered T cells against cancer is getting traction. Notice that 3 of these 5 products, they are all for adults and they are orphan disease. However, for children, there are 4 different products. All of them are orphan disease, orphan drugs, if you want. One, the Kymriah, it's an anti-cancer that is for both adult and pediatric patients. Another, Luxturna, it's a gene therapy for a human retinal pigmentum genetic defect, and is also adult pediatric. So the only 2 products that are specifically for pediatric patients are Strimvelis and Zynteglo. Now, one is for beta thalassemia, so it's a gene encoding beta globin. The other one is a treatment with the genetically engineered hematopoietic stem cell, where the gene for the human adenosine deaminase has been introduced, so to replace the missing gene from the patient. Now, the question is why it's so few products for pediatric patients. If you think that so many of rare disease or genetically originated inherited disease are in children, they appear in children, you would expect that the number of products developed will be much higher. So some general issue can be identified for all ATMP. The regulatory hurdles, well, the legislation, the rules, the guidance, everything the EMA and the other national competent authority have in place were constructed based first on small molecules, then on biologicals. And this kind of product, they can still have a molecular identity well defined. Even a protein or a gene by itself can be defined by its sequence. However, when you talk about genetically modified cells or cells, it is almost impossible to define a molecular identity. So something is different and take time to evolve the rules in a way that covers this difference. Then another point is for all ATMP, the complexity of the manufacturing. It's not so easy to produce a gene, maybe not so difficult, but for sure much more difficult than producing a small molecule by chemical means, or producing protein. Again, the recombinant system are now industrially based, so it is possible, everything is there, but it's much more complex and subject to variability than a chemical reaction. Then something that is linked to the, let's say, claim that this kind of product have a long-term efficacy because they repair something inherited in the patient. So repairing the damage will cure the subject for life. However, one rule is that you cannot claim efficacy if you don't show it. And if you claim efficacy for, let's say, 40 years of life, that is much longer of your marketing authorization, and you have to show it, so you can claim that only after 40 years. This kind of issues put a brake on the reimbursement from the agency because they don't know if it's real or not. And the other point that is true for oral ATMP, it is often this approach. It's a product that is produced for a single individual. So it's a targeted therapy with autologous cells, or it's a gene therapy for a rare disease that is by itself, it's a small population. And this will make this product inherently very expensive, which means that they are difficult to develop, difficult to produce, difficult to be reimbursed, and it's common to all of them. What the regulatory authorities did to help this situation and facilitate the, let's say, developers. One issue were the guidance for the so-called good manufacturing practice. That basically it's a system, a quality system that is in place with inspection in all the European countries, where you have to show that your production system, your production process, it's controlled, it's stable, it is consistent. So that something that you produce today, it's identical to something that you produced 6 years ago. Now, with the cells that have a very short shelf life, or product that have to be stored in liquid nitrogen because they are rapidly degrading, a number of logistic issues rise. You cannot produce something like cells that needs to be delivered to the patient in, let's say, 24 hours from the end of the manufacturing process in one country to another country. Okay, it is possible, but takes a lot of logistic, let's say, arrangement, and anything can go wrong. So one point was suggested production decentralized in the hospital. But this goes against the rules of manufacturing, where the qualified person that has to sign for the release of a lot of a drug needs to be on site. Clearly, this is not feasible if the site are 100 hospitals in a country or across Europe. So all this kind of bureaucratic and legal situation needed to be adjusted. So the European Commission produced a guideline that is for GMP specific to ATMP, and that replaces the GMP for general manufacturing. And these were aspects like decentralized manufacture, final product reconstitution at the treatment center, release of auto-specification products, which is a no-no for a normal drug, but when you have a product that is basically stem cell from a patient, which if you lose it, you don't have anything else to give, then there is let's say, reasoned decision from the physician to provide anyway the product to the subject. And the same, how to manage the good clinical practice for ATMP. Well, they are under revision because they need to be adapted. Another big issue is with gene therapy products, which make use of genetically modified organisms. Now, one big discussion was if a genetically engineered cells is a GMO. Well, it is, but then you cannot expect the patient that receive the product to be authorized to go out of the hospital. And there is some kind, or is that considered a release in the environment? And these are legal, but an issue that needs to be solved. Moreover, there is no common regulation on GMO. There is general guidance, and each member state has applied different kind of authorization models, questions. In an effort to help, again, the developer, the Commission produced an effort, including a question and answer document, a good practice guideline, and the common application for license, a template that is being adopted by a number of member states. Plus, there is on the website of the Commission a listing of which authority in each country is responsible for giving you the GMO authorization, plus indicate on which requirement they ask. Now, this is not only an European issue. This is an issue of all the advanced economies that are able both to manufacture and to distribute this product. So in Europe, the orphan drug and the prime incentives may result in an increase of this product. We know that there is about 17 new products in the pipeline. However, most of these are CAR-T products. So are products for leukemia, tumors, mostly adult, and the pediatric population is disappearing, so that high fraction of pediatric product that was evident up to now will disappear. In the USA, there is even more product coming up. There are at least 30, and they are, let's say, helped through the Regenerative Medicine Advanced Therapy, RMAT. That is a decision, a designation that the FDA gives to a specific product that allows fast track designation, that is something similar to the PRIME model in Europe. So there is an attempt from the regulatory agency to facilitate the development of the ATMP. But again, it's a question of market or science. A large proportion of non-infective disease or not communicable disease affecting children are monogenic inherited pathologies. So why there are so few gene therapies for these subjects? Well, the scientific reason, we have seen already the market, but the scientific reason, it's the lack of knowledge. There is not a clear druggable targets, difficulty to administer and to reach the target with your product, a lack of understanding of the human early development. So when I have to treat this kind of disease, lack of validated juvenile animal models, a lack of a constraint that is reasonable in ethical reason, legal reason, on clinical experimentation in children. But all of these make very complex to develop a new therapy for a disease that maybe it's addressed best treating in utero. And that is already almost a no-no, because automatically everything becomes extremely complicated. Being most of them rare disease, then the market is very small and everything stops because industry says, well, if we have to spend, let's say, $1 billion to develop a new gene therapy, and then we have 10,000 patients, it will not be paid. And this is another big issue to develop a new ATMP for children. EPTRI is an attempt to have a concerted action to address at least the scientific hurdles regarding pediatric drug development. We want to create a research infrastructure that connects and coordinates to access to the pediatric specific resources in Europe and support pediatric drug development with what? Well, excellent science, a quality methodology, and most of all, to remember to everyone that children are a worthwhile target for new therapy. Thank you, everyone.
Thank you, Giovanni. Thank you for having clarified us this really promising and complex reality of the ATMP. Now, the next speaker was supposed to be Evelyn Jacques-Gren from Assistance Publique Hôpitaux de Paris, but for an unexpected urgent commitment, she's not able to join us today. She will send us a recording of her presentation and we will share it on EPTRI website. I would like to thank all the speakers of this session for their interesting contribution. I will now open the floor for the discussion. All the speakers can unmute themselves, so you can comment. And for all the attendees, you can type your question in the dedicated box. Okay, I see there is one question for Pierluigi Martelli.
Yes.
I don't know. Okay, Pierluigi, can you hear me?
Yes, I can hear you.
Okay, there is a question for you. I will read for you. In sequencing analysis, do you take into account the non-coding portion of the genome that regulating protein coding gene may impact on diseases?
Yes, of course. This is a very important issue, and it is starting to be addressed because, of course, the genetics discovered several mutations, several variations, also different type of variations that are duplication, for example, that affect specific diseases in proteins. So a mutation that affect proteins that are involved in diseases. But this is the easy, if you want, part of the problem, because proteins need to be expressed, and all the expression mechanisms involve all the maybe 90% of DNA that is not coding for proteins. But we have a few data at this point, few experimental data that that imply the relationship between the variations of non-coding regions of DNA and diseases. So, of course, this is a very important, very relevant issue. We need data and we need models to try to make hypotheses on the fact that, for example, a mutation occurs in a transcription factor binding site and then can affect the interaction between a protein and the DNA, and then can therefore affect the expression of the proteins that is involved in some pathway, so can hamper the correct working of the pathway. So we need experiments on one side and integrative computational methods on the other side in order to build up a system that can try to make at least hypotheses on the effect of variations. I don't know if I answered the question.
Thank you, thank you. Yeah, I think yes, otherwise, of course, it is always possible for the attendee still writing and maybe ask for further explanation. Okay, if there are more questions, you can write them. Of course, all the speaker and all the presenter are invited to comment or add comment or also question for the other presenter. Okay, there are no more questions. Maybe I have just a really generic question for all the presenters. You have described so many innovative approaches, technologies that can be really useful from omics, but we also talked about biomarkers, in silico models. How we can translate this innovation in the pediatric fields, because we know that the pediatric population is a different population, and so sometimes it's difficult to apply these technologies to this population. So maybe your thought about how we can translate this innovation, how we can push these innovative approaches also in the pediatric field. All the presenter can unmute themselves. Okay, let me check.
Maybe I can give a non-expert opinion, in the sense I'm not in the pediatric field. I'm not even a medical doctor, a physician. I'm a physicist, so I'm not I'm not so aware or not expert in the differences between the adult population and the pediatric population in this field. But I think what we need is, first of all, data. If the population are different, we have to collect data about the differences in the populations. So we need maybe sequencing omics, non-invasive omics on the pediatric population, so to collect data to be contrasted with the data of the adult population, and so to understand what are the differences in the pathway, in the regulations, in the proteins that are expressed, and so on. Of course, also the pediatric population must be stratified in different ages, I think. So first of all, we need data, in my opinion, samples, and then we need to build models that are different for the different populations. In this way, we can have really the idea of what is common and what is different, and also at the level of the therapies, at the level of the insurgence of diseases and so on.
Can I react to this question?
Yes, yes, please.
Thank you. Thank you for the question. That is the basis of the work that we have tried to perform by many years in make evident the differences in the pediatric population and the adult population. I would like to clarify 2 points. The first one is that many differences have been studied and are already known. This is the case of the fundamental pharmacological and toxicological result in
is a major problem and can be solved or at least progress a lot years before the understanding of the variability of gene expression and developmental pharmacology. So pharmacokinetic data are of major importance for the treatment of pediatric diseases in the actual time.
Thank you, Emmanuel. Okay, since if there are no other comments, since we are a bit late, I will move forward to the last presentation, and then maybe we can just have a final comment in the end. I will present the last presentation of the day. I will try to give you a short overview regarding the collaborative efforts needed to promote innovative drug discovery for children. So give me a minute. Okay. So we have already discussed it about some points, so I will go more fast on this point. Of course, drug discovery and development advances in recent years have allowed to find a treatment for many preventable diseases. However, we know that too often children are excluded from these progresses, since most of the new medicines have been discovered and developed for the adult population. And we know that children cannot be simply considered sides down adult, as we were saying just a few minutes before. Many reasons can be mentioned to explain this that have been already discussed. How we can overcome this lack of medicine for children. Experience show that this barrier can be addressed by utilizing a structured approach to research. A structured approach will allow us to overcome fragmentation of pediatric expertise, will provide a consistent framework for all stages of drug development, will speed up research by using the same system and processes for each study, will enhance quality and ensure that the voice of patients and caregivers are heard. Moreover, we need a specific approach at the same time, specific for pediatrics, because of the factor we mentioned, so the impact of ontogeny on drug development, and because also most of child health services are hosted in dedicated facilities and provided by specialized staff. This structural model should include 4 components: context, resources, coordination, science. Context, what I mean is that regulation, drivers and incentives to make drug development appealing to investors in commercial and public sectors, including a clear path from approval to market. Of course, we need resources. We need coordination to avoid the duplication and unnecessary gaps, to promote collaboration between all the parties involved, and to ensure patients and caregivers involvement. And we need science, so rigor, good guidelines and pathway, good techniques leading reliable, repeatable result, coherence across each step of the pathway. As regarding the regulation, we know that regulation introduced since 2007 in both Europe and United States and so to overcome these challenges and highlighted the need for the pharma industry to study their drugs in pediatric population. You have mentioned here the 3 main regulations in question, so the Pediatric Medicine Regulation and Best Pharmaceutical for Children Act and the Pharmaceutical Research Equity Act, made permanent in the United States in 2016. These regulations have incentivized and mandated drug companies to conduct pediatric clinical trials and to expand the labeling to include pediatric indication. Sorry. And moreover, they have stimulated the research on off-patent drugs used commonly in children, have encouraged the establishment and consolidation of investigators' network and trial sites for pediatric research. But as also highlighted in a review after 10 years since the pediatric medicine regulation came into force, although there had been an increase in medicine approved for children in many therapeutic areas, limited progresses have been made in diseases that only affect children, or where there are significant pathological differences between adults and children with the same diseases. And actually, this outcome is not really surprising, considering that the United States and Europe regulatory legislation primarily talks about pediatric clinical trial as an add-on to studies in adults. But pediatric studies should not be afterthought. They should be considered at the beginning of the development process. As regarding the second point I mentioned, the resources, many relevant resources, organization, networks, and research consortia are involved in pediatric research and demonstrated the ability to provide excellent research capacity in the whole pediatric medicine field, including basic research on human development and many topics related to preclinical and translational pediatric research. In particular, in the periods from April 2018 to November 2019, a large survey has been performed within EPTRI, addressing the pediatric scientific community. More than 1,000 key scientists potentially interested in providing services for pediatric drug development were identified, were invited to participate to the survey. And a total of 259 institutions, often grouping more than one research group, distributed across 29 countries, answered to the questionnaire and described the specific field of activities, scientific competencies, and available human resources, tools, facilities, and services they are able to provide. In addition, an online survey has also been conducted to identify the nature and the extent of the need of the user for services in pediatric research, addressing the potential user community, as already discussed by Donato Bonifazzi in the beginning of the webinar. The target user community identified, as we can see here, is mainly composed of academic researchers working at university and hospital. It includes many users from basic research institute or researchers involved in pediatric clinical issues closely linked to the preclinical and translational research. And of course, we need science, what we are talking about today. Many advancements in the pharmacological field derived by the introduction into the medicinal development process of innovative methods and technologies can be mentioned. This is a list, not an exhaustive list. We have discussed today about omics, about in silico method, about biomarkers. But what I want to point out is that relevant advancement took place, but at the same time it is required the adaptation of this innovation, of competencies and methods tailored on pediatric need. Prior to the market launch of a new drug, developers have to test their drug candidate in preclinical and clinical trials. So, for example, preclinical tests require well-established in vitro and vivo models specific for pediatric diseases. Therefore, we need to establish a more effective system to recognize promising basic research at an early stage, as well as to safeguard and develop the results effectively. Even if progresses have been made in recent years from the regulatory point of view, from the science point of view, and as I have said, there are many competencies and expertise in pediatric medicine's research field, relevant structural gaps still exist. Some transnational research infrastructure that aim to support specific activity have been created, but have not worked in pediatric since they lack the specific expertise and facilities for pediatric. To cover these gaps and to allow the pediatric scientific community to become sustainable and well coordinating, the setting up of a research infrastructure completely dedicated to pediatric has been identified as a fundamental pathway. This new approach should include two main aspects: a focus on translational research-driven studies to allow good ideas born in academic and non-profit laboratories to be implemented and therefore became attractive also for commercial sponsors for further development. And the second point, the increase of the public and private partnership opportunities to avoid blocks or failure on the market. Based on this consideration, EPTRI has been proposed as a new infrastructure aimed to accelerate and expand the pediatric drug discovery and development processes by focusing on basic and preclinical research method and taking into account the variability in drug disposition, disease target and disease biomarker, the natural condition of the child growth, requiring size adaptation of medicine form and test, and the advancement in life science, technology, and data science to be implemented in the pediatric setting. To meet the need of the research community, it is important to set up close scientific collaboration and international partnership, as well as to pool financial and human resources also on a global scale. Collaborative approach in the field of pediatric research is indeed at the basis of EPTRI, that has already developed several measures to liaise with the many pediatric initiatives conducted at international level with the aim to integrate a global pediatric research framework. A global outreach and networking approach to pediatric research will increase the impact of EPTRI services and initiative and its global partnership, and will reduce the barrier between basic science and citizen well-being, providing evidence of the direct benefit to society. The key to the success of these activities will be the critical involvement of all the stakeholders, all the parties, so patients community, scientific community, government, the other European research infrastructure, pharma and biotech industries. And while the specific role of EPTRI, as I said, is to aggregate and reinforce the scientific component focused on basic, preclinical and translational research, EPTRI is also aimed to underpin the clinical research phase. To this aim, functionally operational links have been established with, for example, with C4C, the Pan-European Network for Pediatric Clinical Trial, and also with the other many specialty networks and research consortia involved in pediatric clinical research. Collaboration have been also foreseen in the field of rare disease initiative, considering that most rare diseases affect children. Other links have been foreseen with the European Joint Programme on Rare Disease, and also member of the European Reference Network, especially focused on pediatric disease, are also involved in EPTRI activities. Another important initiative I want to mention is that EPTRI will liaise with the ETCC Pediatric Preclinical Proof of Concept platform, aimed to develop a large-scale platform comprising 400 novel research tools based on cells and tissues from patients, covering 10 common childhood cancers. Okay, I would like to conclude saying that from the very early planning phases, EPTRI has a collaborative nature. Its vision is that pediatric research should have a single identity that represents and integrates all the competencies and the initiative of medicine development from bench to bedside, having always in mind the final goal of children's health. In this context, EPTRI will facilitate the translation of the acquired new knowledge and scientific innovation into pediatric clinical study phases and medical use, and will enhance innovative technologies useful to accelerate pediatric drug development processes using an original scientific approach that is pediatric patient-centered. Thank you for your attention. Okay, let me check if we have more questions. Okay. Of course, if there are final comment by the presenter, you are welcome, so you can unmute yourself and add the comment on the last question. Okay, so if there are no more comment or question, we can close the webinar. We are perfectly on time. Of course, you can send us any other request of information or question, and we will try to provide you an answer also in the next day. I would like to remember you that a recording of the webinar will be available on the EPTRI website. And also the presentation of Evelyn Jacques-Gran will be shared in the event section. So, okay, I think we can conclude this webinar. I really thank you for attending the meeting, and I wish you a nice afternoon and a lovely weekend. Thank you.